Hello, and welcome back to biology. So, today we want to talk about multicellularity, right? And one of the big deals that happens is that cells learn to work together, which means that they specialize, which we've talked about before when we talked about differentiation. So, how does this work? How does it happen? And so, there are a couple of things we want to talk about. The first is the evolution of the eukaryotes. And we've talked about this a little bit, but I just want to review. The earliest life were prokaryotes, and we know that they're abundant by about 3.5 billion years ago. Okay, remember, they're single-celled organisms. They have no internal membranes. They have no nucleus, although they do have DNA, and they have very simple genomes. Most of the time, they're actually circular, and they're pretty cool. There are lots of prokaryotes. The biological mass of prokaryotes is at least 10 times that of eukaryotes, and as a matter of fact, you have more prokaryotic cells on you than you have eukaryotic cells with all the fauna that live on your skin and on your membranes and in your gut. So nobody really knows how many species of bacteria live now. I mean, a gram of soil can actually contain up to 4,000 different species. Seawater, right, which is so rich in all kinds of things, may contain as many as 20,000 species. And so there really could be millions of species of bacteria. The problem is that bacteria, many of them are impossible to culture in a lab. And so or you can grow them. Well, I meant culture. Sometimes you can grow them, but you can't keep them growing, which is the culturing. Sometimes you can't even culture them, right? You can't even get them to grow in a lab situation, and so it's very difficult to know what we're talking about when we talk about numbers of and types of prokaryotes. But we know that the prokaryotic domains are the bacteria and the archaea, and the archaea are the extremophiles, the ones that live in like ice cold or radioactive waste or super hot stuff. Now, fun fact, you'd think that both of the prokaryotes, the bacteria and the archaea, would have a common ancestor, but it turns out that the common ancestor is actually between archaea and eukarya. So archaea and eukarya are more closely related than archaea and bacteria, even though these are both prokaryotes. All right, so eukaryotic cells, we know all about those already. They have a nucleus, they have membrane-bound organelles. The genome is really large. The DNA is typically linear and has introns. We'll come back to that later on, not today, but in a different class. And then we, of course, have the cell specialization that's controlled by strict regulation. Okay, the earliest eukaryotes evolve around 2.5 billion years ago, so prokaryotes rule the roost for about 1 billion years. Now, many early eukaryotes were similar to amoeba, protozoa, and unicellular green algae. Okay, now here's your other fun fact. Multicellular life forms appear about 600 million years ago, about 1 billion years after eukaryotic cells evolve. And so it takes about 1 billion years for eukaryotes to show up, and then another 1 billion years or so for them to start working together, because that's what multicellularity is. It's eukaryotic cells working together. So, how do we get these eukaryotic cells? Endosymbiosis. We talked a little bit about endosymbiont theory, but there are two things that happen for eukaryotic cells. Mitochondria probably start out as some energy-producing, free-living prokaryote, maybe a, purple bacteria or whatever, and they are engulfed, actually infect another cell. But it turns out that the energy-producing cells make ATP that can be used by the larger cell, and then they're protected by the larger cell. These smaller cells are protected by the larger cells, and so you develop this symbiotic relationship. Chloroplasts, same thing, except chloroplasts were probably a cyanobacteria of some sort, and they infect, right, and then they're folded in. And the big clue to this is the double membrane. Okay, so if you look in the picture, you can see here's the little bacteria coming in. This is actually here's the mitochondrion coming in, right? Here's the is heterotrophic. Here's the photosynthetic. They come in, the membrane folds around them, and it gives them that second membrane, because they were already cells to begin with. They were prokaryotic cells. Then they become double enfolded. Okay, so just really quickly, what is symbiosis? Two or more species interacting with each other, two or more organisms living closely with one another, two or more species engaged in a long-term beneficial relationship, two or more species interacting in a long-term obligate relationship, or two or more species living closely with one another. Well, it can't be E or A because that means that you would be symbiotic with your dog, and you're not. Okay? It can't be B because that means that you would be symbiotic with your family, and you're not. So what we need is a long-term relationship. Now here's the thing, it doesn't have to be beneficial. As a matter of fact, parasitic relationships are harmful to one of the participants. So what you have is a long-term obligate. An obligate sounds like it, is what it sounds like. Obligated, right? So they have a required relationship. They can't do anything else. They are relying on each other to survive. Okay, in close proximity. Okay. So anaerobic bacteria evolve into mitochondria. I'm sorry, I think I just said anaerobic. I meant aerobic. Bacteria evolve into mitochondria. Cyanobacteria evolve into chloroplasts. Now, plants also have mitochondria, so they actually get both of them. But what is the major difference between mitochondria and chloroplasts? Chloroplasts are green, mitochondria are not. Photosynthesis occurs in chloroplasts, respiration occurs in mitochondria. Photosynthesis occurs in mitochondria, respiration occurs in chloroplasts. There is no major difference. Mitochondria are found in animals, and chloroplasts are found in plants. Well, it turns out that there's actually different colors of chlorophyll, so A is kind of right out. Photosynthesis does occur in chloroplasts. Respiration does occur in mitochondria. So B is correct, C is wrong. That's a huge difference. So D is wrong. And we know for E, mitochondria is found in plants and animals. All right, so evidence, okay, they both have their own DNA and they can regulate some of their own processes, just like the rest of the cell with the nucleus, right? And this double membrane, okay? Now, genes have been transferred to and from the nucleus and the chloroplast / mitochondria. That's called horizontal gene transfer. So it's really kind of hard to establish a clear phylogeny, and we'll talk about phylogeny in a couple days. But what we're talking about is an evolutionary relationship. And so we use genes to track lots of relationships. We can't do it between the mitochondria and nucleus and the chloroplast and nucleus because of that gene exchange, okay? And yeah, there's your fun fact. Okay, so the origins of metabolism, we're just going to do this really quickly, okay? The first prokaryotes were probably chemoheterotrophs, which means they broke down molecules, right? And compounds, and that's how they got their energy. And so they were able to acquire nutrients really easily as long as those nutrients were present. However, when they're not present, they can't acquire them, and ATP was probably among those nutrients. Now, phosphorus is limited, and it's essential, okay? The P in ATP is phosphorus. And DNA and RNA have phosphorus backbones, right? So the skeleton, those side strands, and we'll look at that when we look at DNA, those are all phosphorus. Now, ATP easily stores and releases energy. It's a little energy molecule. It's like a little Red Bull for your cell, okay? And ATP in the environment would decline as populations of prokaryotes increased, and if nobody's making it, there isn't any more. So selection would favor prokaryotes that can synthesize their own ATP, right, which would be the chemoautotrophs, right? So they could take in ADP and phosphorus, an extra phosphorus, put a little energy into it, and make ATP. And actually, they would be maybe doing something else to make the ADP, which we're not going to talk about, but there are other things to do there, okay? Photosynthesis, kind of the same way. Abundance of organic molecules is going to become a problem. Organisms that are capable of making their own organic molecules would have the advantage. They would be selecting for, okay? Light-absorbing pigments, they're called chromophores, would be used to absorb light energy in early photosynthesizers. That's going to develop into chloroplasts, okay? And prokaryotes with the machinery to use water would have a selective advantage. By the way, oxygen is a byproduct of that, and photosynthesis evolves before respiration because there's not enough oxygen in the environment for respiration to occur, and so it's photosynthesis that actually makes that happen. So, and cyanobacteria were probably the first photosynthesizers. So photosynthesis evolves before the eukaryotic plant cells, okay? The first eukaryotes were protists. They're single-celled organisms. They have simple filaments. Today, however, most eukaryotes are multicellular. having said that, all unicellular eukaryotes are in the kingdom Protista, and so when you have these guys, these are all going to be single-celled, okay, the protists. We're not really that interested in protists because what we're interested in is animals, right? Because we are animals, and that's pretty exciting. But really quickly, let's talk about viruses. Viruses, there's a debate as to whether they're alive or not. They do have DNA, but they don't always have complete genomes, and so that could count against them in terms of, is it alive or not? Viruses will infect bacteria, and then they need their host cell, or actually anything. They'll infect cells, and some viruses hijack the cell's machinery to make more of the virus, which means that they're kind of obligate parasites. And so it's hard to kind of say whether they're alive or not. I mean, they do replicate, they do have genetic material, they do evolve, and they do occupy a specific ecological niche. They exhibit variability, but they don't replicate independently, they don't grow on their own, they don't take in nutrients on their own. So they do some of the things, but not other things. It's a debate you can have at length at some other time. All right, so what is an animal? It's a multicellular eukaryote. They're heterotrophic, eating other animals or decomposing materials. So you have your decomposers. They have no cell walls. There are connective types of proteins, usually collagen, that are occurring there. They're usually sexual reproducers. Their development of a zygote will occur through expression of regulatory genes. An example of that would be the Hox genes, which we'll talk a little bit about later on. But this is where we start talking about differentiation and specialization. Okay, so eukaryotic animals ingest organic material, inorganic nutrients, nutrients absorbed from the atmosphere, nutrients leached from rock and dissolved in water. Well, I'm just going to tell you right up front, it's organic material. Animals eat stuff, right? Inorganic nutrients and nutrients leached from rock and dissolved in water, that's going to be plants, right? If we're talking about eukaryotes. And some nutrients can be absorbed from the atmosphere. You're talking about some fungi, okay, things like that. So, multicellularity actually evolved independently many times, okay, several times. Interestingly enough, you start in all of these events with a colony. The cells in the colony that are more specialized to do certain jobs start to do those jobs, and the colony thrives, right? And that specialization just continued, and animals that could have these collections of cells that work together were favored, right? And so you had your unicellular protists, then over time perhaps you have this colony, and then they start specializing. So maybe like these guys in the bottom become food synthesizers, these guys become locomotor cells, and eventually you have this collection of cells that's able to reproduce by producing gametes, the germ cells. These of course are the somatic cells that I talked about in the last video notes. So what are some major evolutionary trends? Over the past 600 million years, there has been an explosion of animal, plant, and fungal diversity. Once we have that transition from being unicellular into multicellular, you have this potential for greater complexity and variation, and the development of organs and organ systems allows these clusters of cells to really specialize to niches, right? So they can exploit different foods, they can withstand different environments, all kinds of stuff. It's very exciting, really. And so tissues are going to be the first thing that when we talk about multicellularity that's going to evolve, that's going to be important. And a tissue is simply an integrated group of cells with a common structure that work together for a common function, right? Now, once we have that, we have this evolution of symmetry, right? And so you get these collections of cells that are organized in such a way that makes a lot of sense from development to adulthood. And there are two big kinds of symmetry. There's radial symmetry, as you see here, right? They have a top and bottom, but you can cut them sagittally any way you want, right? Or transversely. No, coronally. Sagittally or coronally. Bilateral symmetry, that means that you have a left and right. That would be people, that's us, and they mirror each other mostly, right? If you look at your organs, that's not necessarily true, but you have two arms on either side. It's very exciting. They're divided into a dorsal and ventral side, an anterior and a posterior side, okay? Bilateral organisms possess something really important, and that's called cephalization. That simply means that they have a head that has sensory organs. And your sensory organs in your head would include things like your eyes, your mouth, your nose, your ears, and your brain puts all of that information together. So then here's symmetry, okay? So we had tissues, then we get symmetry, then we actually get body cavities, which we talked. The formation begins with gastrulation, okay? And the function of a body cavity is that the fluid actually cushions internal organs and protects them from damage. It can also work as a skeleton for things that don't have bones or cartilage, right? Or exoskeletons. It can enable the internal organs to move independently of each other. So like if you bend over, your organs can shift around inside. It's kind of simple that way. And so here, remember gastrulation, we talked about the body cavity is formed in gastrulation. So when we talk about this, not only do we have this diversity of like tissues and organisms, we have that greater complexity, we have the organ and organ systems, but this also leads to changes in reproductive strategies. It leads to development and adaptations to become terrestrial over aquatic, because everything starts out to be aquatic, and then we start to occupy all of these different environments, and a greater interdependence, more interactions among different species, and so you have whole communities of species working together because of this complexity. It's very cool. Okay, so that brings us to kind of like the Evo-Devo concept, evolution development, a blend of evolutionary analysis with developmental biology. And so we start looking at like what causes changes in animals over time. And so there are three big causes: ecological causes, that's species interactions, predation, competition, things like that; geologic causes, so like temperature, moisture levels, whatever; and then genetic causes, and this is going to be mutations and recombination. Okay, so those are the big three things that dictate the progress of evolution. Right? Now, how do we know all of this stuff? I really quickly want to talk about fossils. Fossils are a puzzle piece of the past. It's a preserved remain of an animal or plant, and it's going to be from an earlier era. It's oftentimes an impression or in a petrified state, and that simply means that it's a geologic geologic formation, like a rock. So whatever the living thing is replaced through mineralization and becomes a rock, or it leaves an impression as it decomposes. Right? So how is a fossil formed? In a nutshell, really quickly: sediment. The animal dies, or the plant dies, whatever. It's buried by some sort of fine sediment, volcanic ash, silt in water, and that protects whatever it is from rotting. Then layers build up on top of that, so sediment accumulates and it pushes down. Over time, silica or something like that replaces the calcium phosphate in the bones, and they become minerals, right? So they become rocks for all practical purposes. And that pressure, right, helps. That pressure of the layers on top really helps with that. And then tectonic plates will cause uplifting, and that will push the fossils closer to the surface, and then erosion processes will uncover them. Yeah, and so eventually people will either see them on the surface or they won't have to dig very far to get to them. Okay, so fossils occur in sedimentary rocks. Igneous conditions, metamorphic conditions destroy remains, because you're talking about lava, it's hot, or metamorphosis, pressure and heat, that's bad for preservation. Okay. There are sometimes fossils that will form in low-grade metamorphic rocks. We're not really concerned about that now. And sometimes trees are buried by lava flow, but again, we're not concerned about that right now. Overall, organisms have to be buried really rapidly to prevent decay. That's it. And it's sedimentation that does that. Okay. So which location would yield the most fossils? Well, the Amazon basin is way too moist and way too hot. Things decompose really quickly there, so no. Alaska, really cold, and that would be great for natural mummies, but not so much for fossils. Texas, well, it used to actually be ocean around here. We've had lots of water coming in and out and sedimentation. We have a ton of fossils in the Barnett Shale. We also have oil and natural gas because of some of those very same processes. And in the Sahara, that is a really super dry desert. There's not going to be a lot of fossils hanging out there. Okay. Now, what's most likely to be preserved? Hard parts, bones, shells, exoskeleton, wood, right? Stems, stuff like that. Almost never preserved, right? Internal organs, skin, hair, and feathers. Now, having said that, skin, hair, and feathers can actually leave imprints. You can see on this beautiful Archaeopteryx, this is the transition between dinosaurs and birds. You can see the imprint of feathers, and here you can see a beautiful, very clear imprint of a single Archaeopteryx feather. Very cool stuff. Okay, so which of the following animals would be most likely to be fossilized? A crab, a jellyfish, a beetle, or a butterfly? Well, going on the hard parts thing, that would be the crab. Jellyfish, imprint maybe. Beetle, it's got a little bit of an exoskeleton, but it's fairly brittle. Butterfly, same thing. Okay, might get an imprint, but crab would be the one that would be most likely to be fossilized. And the main types of fossils are petrified fossils. These are bones that have had the bone part replaced by other minerals. Molds and casts. A mold is where you have the thing is buried in sediment, and then for whatever reason it goes away and you're left with a void that is the shape. It's a mold, like in ceramics class, right? It's the same thing. And then casts are the part that was inside. And sometimes what you'll get, and that's really cool, is you'll get a mold fossil with the cast still inside, and when you break it apart, you've got the mold and the cast. It's very neat. Okay. Carbon films are when living things, mostly plants, get compressed and decompose. They leave well, they don't really decompose, so that's not important. They leave a trace, a darker color on the rock. So the thing itself is not there, but in a way it's almost like a stamp, you know? Okay, and then trace fossils are going to be things that are signs of living things, so footprints, ripples, coprolites, which is petrified poop, things like that. And then lastly, actual preserved remains like insects in amber. Okay, and then just really quickly, how do we know how old things are? We do some relative dating, which would be the layers of the rocks, right? And then we do some radiometric or absolute dating, where we actually look at radioactive decay of various elements in the rock to get actual numbers, and then the layers themselves. So we know that this layer is older than this layer, right? And then if we date this using radiometric, we know that this is 450 to 850 million years old. This one has to be older than that. All right, and biogeography is kind of the study of these fossils in relation to where they were on Earth. And I put this in here because it's actually really neat. A lot of the fossils that come from 250 million years ago, you can actually find on separate continents, because that's when we had Pangea. Everything was a single continent. So the animals that evolved then, or plants that evolved then, actually can be found all over the world because now the continents are far apart from each other. All right, so fossils really do indicate plate movements over time, and that's a very helpful thing in addition to just rock structure. All right, so both the images in the previous slide indicate that the continents have always been where they are now. South America used to be close to Africa. Ferns can be found in Antarctica. India was south of Asia and unconnected to other landmasses at one time. Well, the picture shows that Africa and South America used to be close to each other, and then they split apart, forming the Atlantic Ocean. So here's your fun quiz. Success or failure? Dinosaurs extinct, but they lived for 150 million years. Recorded history, 5,000 years. For every year of recorded history, dinosaurs had 30,000 years. For every day of recorded history, dinosaurs had 82 years, and for every minute of recorded history, dinosaurs had 3 weeks. So, it's not always a judgment of success that you're extinct. Okay, now obviously in the grand scheme of things, you know, it's a fail if you go extinct, but by the same token, you'll also have to look at how long a run did you have, right? Dinosaurs, 150 million years. Do we think we're going to get that? I don't know. Maybe. We'll see. All right, so that's it. Thanks for listening, and I will see you next time.